undifferentiated cells present in living systems which have the enormous capacity to multiply and develop into many different cell types. They possess an explicit ability of balancing between self-renewal and differentiation into new cells. Stem cells can divide indefinitely, whereas the differentiated cells in the tissues and organs of the body cannot. One of the very important roles of stem cells in adult organisms is the internal repair mechanism. Proliferation of stem cells may yield two identical daughter cells, which are also the stem cells, that is undifferentiated cells, or one stem cell and another a bit differentiated cell with specific functions. The undifferentiated cells resulting from the proliferation of stem cells are capable of self-renewal. The differentiated one is a precursor or progenitor of some specific lineage. This is how stem cell replenishes itself and also forms differentiated cells. Life starts with a stem cell. Fertilization of an egg leads to the formation of a zygote. Multiple mitotic cell divisions lead to the formation of a blastocyst. Inner cell mass of the blastocyst is the embryoblast and the outer cell mass is known as trophoblast. The outer cell mass is also known as trophoectoderm, which forms placenta, chorion and the umbilical cord. The inner cell mass ICM forms the embryo. Under the influence of appropriate growth factors, these cells differentiate to generate progeny, leading to the formation of three germ layers: ectoderm, mesoderm and endoderm. These cells can be maintained in cultures as stem cells by providing suitable conditions. Stem cells culture study is the field of interest because of its potential to unravel the details of development of diseases and production of therapeutics for treatment of diseases. Work on stem cells started somewhere in 1960s. Characteristics of stem cells: they originate from a stem cell population, they exhibit the ability to self-renew indefinitely in the undifferentiated state. They maintain normal karyotype during growth. They can differentiate into all three embryonic germ layers. Classification of stem cells. Stem cells can be classified according to their plasticity into totipotent stem cells, pluripotent stem cells, multipotent stem cells, adult stem cells and progenitor or precursor cells. Totipotent stem cells. A fertilized egg is a totipotent cell. It can form any cell type of the body and can give rise to a complete functional organism. The cells which develop during first few divisions of the zygote are also totipotent cells. They possess highest degree of plasticity. Pluripotent stem cells. These cells developing after 4 days in the embryonic cell division are the pluripotent stem cells. Embryo develops 2 layers after 4 days. The inner layer cells are pluripotent and can give rise to all body tissues, but not without the help of the outer cells. The inner cell mass ICM is harvested for various stem cell applications. Multipotent stem cells. Multipotent stem cells are less plastic and more differentiated. They can form a limited type of tissues. They are derived from pluripotent cells and are the progenitor of certain cell lines. These cells have the capacity of self-renewal and can form any type of cell for some specific tissues, for example, mesenchymal cells. Adult stem cells. Adult stem cells are also the multipotent stem cells. They are the undifferentiated cells present in differentiated tissues in organisms. Differentiated cells need to be replaced in mature adult individuals also, for example, in blood, connective tissue and nervous system. Their presence has been identified in many different tissue types such as hematopoietic, neural, endothelial, muscle, mesenchymal, gastrointestinal and epidermal cells, progenitor or precursor cells. The adult tissue contains stem cells for replacing the worn out cells. These tissues also contain partially differentiated cells known as progenitor or precursor cells. Stem cell sources. There are various sources of stem cells such as embryonic stem cells, human embryonic germ cells, induced pluripotent stem cells, adult stem cells, cord blood. First, embryonic stem cells. Embryonic stem cells are derived from the totipotent cells which are present in the inner part of blastocyst. They can develop into any of the three germ layers, ectoderm, mesoderm or endoderm. These cells can be harvested from inner cell mass after 4 to 5 days of in vitro fertilization. They exhibit indefinite self-renewal capacity in vitro, but may undergo some changes in the chromosomes after good number of passages. Human embryonic germ cells. They are derived from germline cells of early fetal tissue. Embryonic germ cells can give rise to 3 germ layers and all the specific organs of these layers. These cells are further in development than the embryonic stem cells and can give rise to limited types of cells. Induced pluripotent stem cells. They are the somatic cells which are reprogrammed back to the pluripotent cells. They possess the potential to differentiate into any cell of the body. This is done by introducing certain genes inside them such as SOKM and SOLN combinations which helps them acquiring pluripotency. Adult stem cells. Adult stem cells are the undifferentiated cells present in several differentiated tissues. These somatic stem cells are present throughout the body. Their major role is regeneration and repair of the tissues. They exhibit limited self-renewal capacity in the culture conditions. These cells are also referred as multipotent cells. These cells are comparatively of lesser use for disease treatment purpose. Confirmed capacity of these cells to differentiate into a specific cell type draws the attention of researchers towards them. Some of the methodologies have reached clinical trials also. Cord blood. Cord blood is a rich source of hematopoietic stem cells. Cord blood is collected at the time of childbirth. It is the blood which is present in umbilical cord after the child's birth. It contains large number of stem cells which can rapidly multiply. Presence of stem cells in adult organisms. All the parts of the body contain some stem cells. They are present in bone marrow, muscles, baby teeth and hair follicles, etc. Stem cells present in adults are the multipotent cells, for example, mesenchymal cells, neural cells and hematopoietic cells. Mesenchymal stem cells are found in most of the body tissues. Mesenchymal stem cells are heterogeneous group of stem cells capable of differentiating into multiple cell types including adipocytes, chondrocytes, osteoblasts and cardiomyocytes. Neural stem cells are present in adult brain for replacing the worn out and dead cells. Hematopoietic stem cells also possess multilineage potential as they can differentiate into all blood cell types. Mesenchymal stem cells are the most commonly used cell types in clinical trials for stem cell therapy. Culturing of stem cells. Stem cell culture is attracting great attention because of the possibilities of treatment of diseases which otherwise have no cure. An important concern with stem cell culturing is the requirement for developing appropriate culture conditions which will enable the cells to maintain its characteristics of self-renewal and pluripotency both. Stem cells can yield millions of cells in the laboratory. They can retain normal karyotype when cultured for long time, unlike differentiated cells which tend to develop aneuploidy and malignancy. Stem cell culture parameters are critical and require a medium which should be serum free, xeno free, chemically defined, and should be able to support the growth of all types of human pluripotent cell types. Culture conditions for stem cells vary with the type and origin of cells. Medium conditions also depend on the purpose of the stem cell culture. The microenvironment required for studying some cell processes shall be different, and for the production of therapeutics shall be different. The oxygen pressure required by stem cells is lower than the atmospheric level as it grows at a lesser pressure in vivo. Due to continuous metabolic processes taking place inside the cell, its microenvironment constantly changes. Stem cells are growing in a specialized three-dimensional microenvironment inside the body known as stem cell niche, which is very difficult to simulate in vitro conditions. Stem cells grow on the culture medium as tightly packed colonies. The cells have high nucleus to cytoplasm ratio. Human embryonic cell lines express high telomerase activity, whereas the somatic cells possess shortened telomerase and no telomerase activity. Telomerase expression is highly correlated with the immortality in human cell lines. The somatic cells enter into the senescence and have a finite lifespan because of no telomere activity. Embryonic and germ cells exhibit high telomerase activity, which enables them to multiply extensively. Embryonic stem cells exhibit three basic properties. They replicate indefinitely without undergoing senescence. They replicate without any change or mutation in the genetic material. They can differentiate into various cell types such as neurons, blood cells, cardiac and skeletal muscles, etc. Isolated embryonic stem cells grow extensively in the culture medium flask and give rise to millions of such cells. These cells are a valuable source for research purposes. These cells retain their pluripotent character even after 4 to 5 months but do not show any malignant properties. Critical concerns during stem cell culturing, that is, contamination, cell line stability, cell line authenticity. Contamination. Culturing of cells in laboratory must be carried out in aseptic conditions. Contamination with bacteria, fungi, viruses and mycoplasma is a common problem. It causes cell death and the loss of entire culture. Human stem cells are sensitive to microbial contamination as they are grown in enriched medium without antibiotics. Contamination with viruses can alter the properties of cells at genetic level. Cell line stability. The cells continuously growing on artificial medium tend to undergo changes in the genetic makeup, which can also be observed phenotypically. The chromosomal abnormalities in the cells can be detected by the karyotyping methods. The stem cell characters must be detected regularly while culturing them, because once they lose these characters, their applicability finishes. They must be able to give rise to any of the three germ layers. Cell line authenticity. Cell cultures may be cross-contaminated in the laboratories because of a variety of cells being cultured at the same time. The rapidly growing and transformed cells may overgrow and contaminate the slow-growing cells. Cell cultures need to be authenticated for their unique features by testing them before conducting any study. Stem cell technology. Stem cell technology has huge scope in research as well as healthcare. Millions of people globally are affected by chronic, degenerative and acute diseases. Development of the stem cell culturing has opened new hopes for finding treatment for the diseases which have no cure as of now. Major diseases like cancer, type 1 diabetes in children, nervous system diseases, primary immunodeficiency diseases, diseases of bone and cartilage are being dealt with. Cell transplantation has been successful in treatment of extensive burns also. Good amount of research has been done on mouse stem cells. The information regarding human stem cells is also somewhat similar to it. It was observed in 1960s that certain mouse cells can give rise to various different types of cells and tissues. Stem cell therapy for some diseases is already in use, such as bone marrow transplant for cancer patients. The blood stem cells, which can give rise to all the cell types in blood, are given to the cancer patients to restore the destroyed cells because of chemotherapy and radiotherapy. Elaborate technologies have been developed for large-scale growth of human pluripotent stem cell cultures in last decade. Adult stem cells, also known as multipotent stem cells, are the best source for therapies, although they cannot match the pluripotency of the embryonic stem cells, but they can be used for development of limited types of cells. Multipotent stem cells can differentiate into bone, cartilage, muscle, fat or other tissue types. Their use for bone and cartilage replacement is undergoing FDA approved clinical trials. Adult stem cells can be taken up from the adults who have to be treated and can be differentiated into healthy tissues. These can then be transplanted back into the patients for treatment. This is referred to as autologous transplant. This method does not involve any ethical or legal issues. There will be no chances for viral or bacterial cross-contamination from other individuals. Allogenic transplant is also practiced clinically. Hematopoietic cells are of much medical interest because they have narrower range of differentiation. They have been successful in treatment of cancer. Although these cells are advantageous for developing desired cell types for blood, but the use of adult stem cells cannot serve the purpose for all kinds of therapies. Applications of stem cell technology. Human embryonic stem cells have emerged as a potential tool for cellular therapy and regenerative medicine. Their ability of self-renewal and to differentiate into cells of various lineages can be applied for treating numerous diseases and disorders. Cord blood banking is becoming popular with increasing awareness about stem cell benefits. Cord blood contains high percentage of stem cells which can be collected at the time of childbirth. They can be preserved for long period of time and can be used later to treat lots of diseases. They have proved quite useful in clinical applications. Cord blood banking allows the treatment of patients with their own stem cells. These cells can be used to treat more than 80 different types of diseases, which can be categorized into following groups: cancers, blood disorders, metabolic disorders, and immune disorders. Cancers: leukemia, Hodgkin's disease, non-Hodgkin's lymphoma. Blood disorders: aplastic anemia, thalassemia, sickle cell anemia. Metabolic disorders: congenital neutropenia, DiGeorge syndrome, severe combined immunodeficiency diseases. Immune disorders: Hunter syndrome, congenital erythropoietic porphyria, Wolman disease. Clinical trials are going on for the treatment of cerebral palsy, autism, hearing loss, and spinal cord injury. Several major diseases have been treated by stem cell technology, such as type 1 diabetes in children. Type 1 diabetes, an autoimmune disease, occurs because of the destruction of insulin producing cells. They are destroyed by the autoantibodies produced by patient's body. This problem can be treated by transplanting normally functioning cells on pancreas. Pluripotent stem cells are grown and programmed to differentiate into insulin producing beta cells of pancreas. Successful transplantation of such cells shall treat the patient permanently from such a disease. A company named ViaCyte, based in California, has successfully treated patients suffering from type 1 diabetes by stem cell therapy. Pancreatic precursor cells were encapsulated in semi-permeable membrane and planted under the skin. Nervous system diseases. Nerve cells need to be replenished whenever they are damaged. Damaged nerve cells and their failure to be replaced by normal ones may lead to different types of nervous system diseases. Neurodegenerative diseases occur because of loss of nerve stem cells to replenish the degenerating cells. Replacement with new stem cell serves as the possibility of disease treatment. Destruction of dopamine producing cells leads to Parkinson's disease. Loss of neurotransmitter producing cells leads to Alzheimer's disease. Loss of cells is also observed after spinal cord injury, brain trauma, or heart stroke. Such patients can be treated by placing the normal and functional pluripotent cells in the tissue, which can differentiate into required nerve cells. Surgical implantation of fetal cells in the brain tissue of Parkinson's patients has resulted in the treatment of these patients. Studies are going on for the treatment of Alzheimer's patients by this method. Transplantation of stem cells is considered as one of the ideal methods of treating nervous system diseases. Primary immunodeficiency diseases. Immunodeficiency diseases can be treated by transplanting normally functioning pluripotent cells. Severe combined immunodeficiency disease, Wiskott-Aldrich syndrome, and autoimmune disease, lupus, are some of the complications which can be treated by this method. Diseases of bone and cartilage. Potential use of stem cells for patients who have suffered limb loss or degeneration of bone or cartilage is evolving as a promising therapy. Presence of mesenchymal stem cells, which are non-hematopoietic, is observed in bone marrow cells. These cells can be used as regenerative medicine for bone repair, cartilage repair, tendon and skeletal muscle repair. Cancer. Cancer is a disorder which is characterized by the uncontrolled growth of any type of cell in the body. The origin of cancerous tissue can be from the stem cell, progenitor cell, or a differentiated cell. Cancer cells can form tumor at distant places and invade various tissues by moving from one site in the body to another. This is known as metastasis. Treatment of cancer involves chemotherapy and radiotherapy, which kills the cancer as well as the normal cells. This leads to the loss of active immune cells and the stem cells, which should multiply and replenish the worn out cells. This may render the patient prone to several infectious diseases. In order to maintain the supply of stem cells and the cells of immune system for carrying out normal body functions, transplantation of stem cells can be done. Applications of stem cell technology in research. Culturing of stem cells in vitro helps us to understand how these undifferentiated cells change stepwise into highly specialized and differentiated forms. Example: skin, bone, brain, liver and other cell types. Disorders such as cancer and birth defects are usually due to errors occurring at the time of cell division. These can be unraveled by studying the cell growth in controlled conditions. Genetic and molecular control governing these processes can be helpful in developing therapies for such medical implications. Research is also going on gene therapy to be used as the tool for curing diseases. The normal functional genes are introduced inside the stem cells and transplanted on the defective organs for expression of the missing protein. Conclusion: stem cell science has immense potential of starting a new life and supporting it throughout. Stem cell culturing has increased our understanding for the life processes, and it works as a safe backup for all the living systems.